afternoon and welcome you all to this AICTE ISTE sponsored training on pilot plant scale-up techniques. So in this afternoon session, we have Dr. Vasudevrao Avapati sir with us to have this session. So before I hand over this, I wanted to give brief introduction of sir. Sir completed his bachelor's and master's degree from Andhra University, post which he has completed his PhD under the expert guidance of Dr. Y. Rajendra Prasad sir. He was awarded with CSIR Senior Research Fellowship by Government of India for completing his PhD degree. Sir is a registered pharmacist, young scientist, an expert in the field of design and synthesis of bioactive hybrid molecules, computational drug discovery, with a focus pioneering on hit-to-lead optimizations associated with the development of novel bioactive ligands to fight against multifactorial diseases. To name a few of his achievements, which includes he was the recipient of the International Medical University E-Learning Achievement Award for the year 2019. Sir is an innovator of technology-enhanced learning strategies, and he is a principal investigator of the prestigious FRGS grant, which is awarded by Ministry of Education, Malaysia. In the International Medical University IMU, the Learning Resource Festival Innovasi competition was held, in which Sir has secured gold medal for his e-learning innovation project, which showcased titled Virtual Innovations via Simulation Training and Assessment. In the second Higher Education Leadership Meet, the Venus International Foundation recognized and conferred him with Young Leader in the Health and Medical Sciences discipline. Sir is currently a senior lecturer in the Pharmaceutical Chemistry Department and an e-learning lead in School of Pharmacy at the International Medical University, Malaysia. He developed innovative teaching and learning methodologies for the various course modules based on new drug discovery in undergraduate and postgraduate programs at the IMU, which help students to practice theories and principles of drug discovery and development. So with this, now I invite Vasudevrao sir to kindly proceed with the session.
Testing, is my voice loud and clear?
Yes, clear.
Okay, thank you. So, thank you, Dr. Pramod, for the introduction. Without any further delay, I'm going to share my screen.
Yes, visible.
Oh, is it full screen or
Yeah,
how it's appearing?
it is full screen now, sir.
Full screen. Okay, thank you. A very good afternoon to all the participants of today's AICTE CHIPS induction program. My name is Dr. Vasudevrao from School of Pharmacy International Medical University, Kuala Lumpur, Malaysia. In today's section, I would like to highlight few informations regarding simulation integrated scaling of processes in drug discovery from bench to market. So before we discuss about our actual topic, I would like to highlight a safety concern. Across the globe, we are facing the pandemic situation currently. Still, the combat is not yet over. So please stay safe and take care. So what's the aim of today's webinar? So in this webinar, my main aim is to introduce participants with the information related to Role of simulation for scaling of processes in drug discovery. First of all, the concept of scaling of processes in drug discovery. This scaling of processes may sometimes need step up or step down scaling. Most of the times, we may see the scale up processes because the bulk drug industries mainly deal with that. However, how about the scale down processes? There is equal importance and significant momentum also required for the scale down processes as well. What are the scale down processes and what are the various areas scaling down processes are mainly needed? Let us discuss in detail. So the intended learning outcomes of this presentation includes to explain the strategies and applications of simulation integrated drug discovery with special focus on ligand-based drug design, and also to acquire the knowledge to practice quantitative structure activity relationship modeling and simulation for scaling of processes in drug discovery. So these are the intended learning outcomes of this workshop. I'm going to facilitate in the next few hours, 1.5 hours duration. Okay, these are the webinar contents. I'm going to take you through introduction, simulation strategies, QSR modeling, OECD principles for QSR modeling, application summary references, followed by acknowledgements. So let me introduce to you all a small brief introduction to the conventional process of drug discovery. In the conventional process of drug discovery, we may need to go through several stages, which includes the drug discovery, preclinical and clinical development and regulatory processes. So among these stages, it is not a overnight process. Hardly it may take 12 years of time and 1 billion plus research and development cost in order to get one molecule into the market. So such a tedious process is a drug discovery process. So every stage by stage need a transition, and that transition is the place where majority of the failures may happen. So what type of failures generally we may come across with the drug discovery processes, and additional things we will discuss. So if you look at the US FDA processes, preclinical, clinical and NDA review, altogether it is actually taking around 12 years of time and 1 billion R&D costs. Based on this, what is our main uh, message we can consider? If you look at the timeline, it is first of all very huge pathway, and it's a long way to go. And similarly, on the other side, the cost effectiveness. So it's very costly. At the same time, moving on to the next stage is very complex. So three major things. One is very costly, very complex, and time, very long time. Because getting one marketed drug, if you expect 12 years of time, it's rather very unusual. Because we are moving into different types of pandemic situations. Today, Corona, some other day, some other. So we may need to accelerate the drug discovery process. How the drug discovery process can be accelerated? We need some complementary strategies. So these complementary strategies may accelerate the process of drug discovery. One best example I would like to talk about is that drug repurposing. Drug repurposing is one of the best complementary strategy currently across the globe the R&D scientists are using. Why? Because identifying a new drug molecule has its own timeline and has its own regulatory needs. So the drugs which are already being received approval from the FDA, why don't we test for the new uses for the old drugs? That's what the drug repurposing strategy. So that accelerated the process of drug discovery. Why? Because the preclinical stages and 50% of the clinical stages can be omitted because the safety efficacy has already been tested. So like that, we are looking forward to have better strategies to accelerate the process of drug discovery. Moving on, what is the major challenge? As I mentioned just now, the transition probability is the major challenge. However, time to time, since 1970 to 2010s, the scale of the expenditure associated with the drug discovery, if you leave, it already reached 2.6 billion. So the cost effectiveness and the transition, smooth transition gap should be covered. So what is the best strategy to cover these two? So why you are incurring so much expenditure in this phase? Because you may need to perform so many experiments. You may need to conduct several studies. So you need to reduce the studies, but the standard and quality of the outcome still need to be retained. So how to do that? So simulation has been identified as one of the strategic tools to reduce this gap, as well as to reduce the total number of experiments to be conducted at the laboratory scale. In the title, you could say bench to market. So what is the actual meaning of bench to market? So the molecule, before it reaches to the market as a potential drug molecule, this molecule will be termed as different names. Initially, it is just a chemical. Initially, it is just a chemical. Once it's subjected for any biological assay and identified as a potential active compound, and the moment it has some bioactivity, the compound is identified as hit molecule. And this hit molecule, if it is having desirable properties associated with the drug-like molecule, so this molecule can be considered further as lead molecule. And if the lead molecule has also satisfied majority of the drug-like properties and exhibited some biological potency, so this compound can be further carried to the next stage, that is clinical trials. So it is a clinical candidate and so on. Once the clinical candidate stage to drug. So you just observe here a simple chemical molecule. Once after it enters into the drug discovery pipeline, how the transformation is taking place as a hit molecule, as a lead molecule, as a clinical candidate, and so on as a drug. So what is the main difference associated with this transformation? The main difference is nothing but the properties of a molecule, the properties related to the drug, the properties related to absorption, distribution, metabolism, excretion, and toxicology. So these are all the favorable features we may need to impregnate and integrate into the molecule so that the molecule may be further identified as further scope to move forward in the phases of the drug discovery. Otherwise, if the molecule, although it is a hit molecule, if the molecule is toxic, it cannot be subjected forward to the animal studies, or it cannot get any kind of recognition at the lead optimization stage. So it's all depends on the properties of a molecule. So this is all about the bench to market. So these are very tedious steps and designated requirements to be satisfied by a molecule. Unless otherwise, it cannot be entered into the next phase. So let us talk about the key concept of our today's webinar, simulation. So what do you mean by simulation, first of all? Okay, what British English and American English says about simulation? Let us see a small definition. In under British English, you can say a representation of a problem, situation in mathematical terms, especially using a computer. So simulation means the activity that uses the computer. Okay, construction of a mathematical model, some processes, situation, etc., in order to estimate its characteristics or solve problems about it, probabilistically in terms of the model. So this is all about the mathematical relationship. The background working model is the mathematical model. And similarly, in American English, you can say the use of computer to calculate by means of extrapolation the effect of a given physical process. So the extrapolation of a given physical process. So meaning that simulation is a mimicking process. Simulation is a mimicking process. The real experiment established based on several factors. Based on these several factors, you develop a mimicking model, that is a virtual model, that is simulation. So it mimics the similar process and it will produces the hypothetical result. And this hypothetical result we can obtain based on a computational algorithm. So that's what the math, because algorithm basically constructed with the mathematical relationship. And we can see here the duplication or reproducing of certain characteristics or conditions of a system, physical process, use of a model or representation for studying, training, etc. So based on these two definitions, one thing we could clearly figure out is that simulation is the process of doing a duplicate activity using computer. In simple words, we can say that. So using a computer, if you are mimicking a real experimental process, that is nothing but simulation. So how this simulation is really helps a lot. Let us discuss further. So simulation, there are several landscape. The major strategies are categorized into artificial intelligence, machine learning, and deep learning. Of course, under every strategy, our drug discovery phases are integrated. So these drug discovery phases basically integrated through two means of major branches, like one is bioinformatics and the other one is chemoinformatics. The conceptualization of the biology integrated with information technology and associated algorithmic and machine learning, because every software, the background working is algorithm. So it is all about designing an algorithm that works similar to the real atmosphere. So there are different advancements. I'm not going to talk about all these things because I'm not the software professional. However, these are the advancements which are really complementing and accelerating the process of drug discovery in the current scenario. So just based on the definition, what is our primary understanding is that if the real process is existing, mimicking the real process is the simulation. Okay. So for the real process and mimicking, what is the key bridge? That is data. Without this data, we cannot actually design an algorithm similar to the real experiment. So in order to design a simulation model, we may also need to have the proof of concept data. So that means the data obtained from the real experiment. And this data is the foundation to develop the simulation process. Because simulation is the process of mimicking the real experiment. For example, the number of experiments required to design a compound, maybe thousands or hundreds or thousands of reactions. For example, if you integrate simulation technology into the reaction mechanism, reaction uh, processes, for example, we can actually predict some sorts of reaction based on the quantum mechanics and also the molecular mechanics, because every single algorithm designed based on the natural laws of for example, if you consider the computational drug discovery, artificial intelligence and all, what are the foundations for developing the algorithm? Molecular mechanics and quantum mechanics are the foundations based on the atomic physics and also the Schrodinger's wave equation. And these are the key theoretical concepts established earlier back in the histories in our um, physics and chemistry and all. Based on these theories and empirical relationships, they design the algorithms and they test the model whether the predicted information can align with the simulation data or not. So this is how the data will produce some information, and based on the information we can derive the knowledge, and wisdom is the way you apply the knowledge. And the decision making and the understanding, consolidation and application analysis, these are all the process different types of ways to apply the data. So decision to risk assessment. So the actual context is totally based on the data. For example, you are specifically working on diabetes. So you may need to have the diabetes associated data on a drug target, which is primary target of in the pathogenesis of diabetes. For example, PPAR gamma. So you are designing a simulation model to fight against PPAR gamma or to target the PPAR gamma. So whatever the PPAR gamma associated real experimental evidence is there for from the existing drugs or existing experimental compound, it must be the foundation and it must be the pillar of the whole process of simulation. So this is the direct relationship between the data, information, knowledge, and wisdom. That's the application. So why do we need simulations first of all? As we discussed this few minutes ago, because we need some complementary strategies which can reduce the gap, because where the translational gap from one phase to another phase is rather very high. The rate of success is very, very less when compared to the rate of failure. So how you are going to achieve more success, or you try to balance the gap? So we may need some complementary strategies. However, one thing I would like to highlight here, none of the phases associated with the conventional drug discovery process can be replaced by using simulation process. So simulation process is just a complementary strategy, but not a replacement strategy. It is not at all a replacement strategy. It is just a complementary strategy. That means it will accelerate the process. However, it will try to limit the number of experiments to be conducted in the laboratory. That's all. So once again, whatever the hypothesis that you will generate using a simulation process or simulation model or software or algorithm finally need to be end up with a laboratory experiment. And that laboratory experiment must be the proof of the concept, proof of the concept. So the simulation outcome is just a concept. It's just a virtual hypothesis. And this virtual hypothesis must be tested. So how you are going to test it? Using an experimental method. So that's how we can actually reduce the gap and try to handle the translational failure. And these failures mostly will take place in this translation. This area is called valley of death. Valley of death, where the strategy fails and almost the whole thing is getting collapsed when we are in the process of identifying some new compound as a drug molecule. If the whole thing is collapsed all of a sudden, what about the R&D cost, manpower, time, everything wasted? So this is the place where we may need to address. I don't say simulation is fully addressed this problem at. So no single strategy identified to address this process. If that's the case, by this time we would have received good number of drug molecules to fight against corona, is it not? But unfortunately, so far we could not have a single molecule that is very specific and potent to fight against coronavirus. Technology is so advanced. What could be the possible limitations? Because multifactorial and multiple complex things are actually uh, needs to be handled at different stages of the drug discovery process. However, still we could manage to get one molecule successfully approved by FDA specifically for the treatment of coronavirus disease. What's that? Remdesivir. The remdesivir is also because of one of the complementary strategy developed by our scientists. Which strategy is that? In the beginning of our presentation, we discussed on it, drug repurposing strategy. So that is how the process acceleration and reducing the gap of the transitional failures in this process. So general process of building a simulation model. So in the general process of building a simulation model, as you can see, the starting point is a real system. So without a real system existing, you may not be able to develop a simulation model. Why? Because real system is the foundation, it's like a heart. And from there you can design an artificial heart. Yes. So if you know the functioning of the heart and how it designed and what is the structure, everything, you can actually design a artificial heart. So in the same way, it's a simulation process also mimics the real experimental process. So this is the hierarchical diagram where you can see the experimental results and simulation results can be comparable. And this comparison and further validation, this is called validation. And this validation is the proof of concept. Once the validation is successful, you can reduce the total number of experiments which generally required to perform a screening process. However, still you can manage to gain the same outcome by mimicking the simulation process in the simulation model. So this is the stage where majority of the companies may undergo some typical situation because of several challenges. However, established strategies are well integrated, but every strategy has its own limitations with respect to the factors associated or with respect to the way algorithm works and with respect to reproducibility of the results. So there are several things we can actually talk about. So just I would like to highlight what are the various phases mostly the simulation can be integrated. So the starting from the disease related genomics and until the clinical trials, in every stage, a simulation experiment can be integrated. So the simulation is such a diverse range of tools are available nowadays. Nowadays, diverse range of tools are available. So tools in the sense, what I mean to say is that applications, software applications. So these software applications, because majority of the phases in the early phases of the drug discovery, they are not intended to scale up the processes. Why? Because scaling up processes is required once everything successful and received the full approval for the compound to use it as a drug molecule, then we may need bulk drug synthesis, pilot plant construction, and so on. But however, in the initial stage, the early stages of drug discovery, we need scaled down processes because to limit the expenditure associated with the drug development and to limit the cost to maintain the associated with animal studies and also the absorption distribution metabolism studies and all target validation lead discovery. So every stage has its own strategic endpoints. So these strategic endpoints, how to meet. So as we already discussed in the beginning, it takes 12 years of time and 1 billion US dollars. So what's the complementary strategies we can consider? Several n number of tools are there. I may be introducing to you all few popular tools, majority major strategies. But however, besides to that, several other strategies also available. Let us discuss. So this is just a simple analogy, lock and key type of analogy, where the the specific key only can open a specific lock, something like that. Based on this, the receptor theories and also the selectivity and specificity of the compound design, these all things actually developed. So this is how the integration of simulation strategies into the drug discovery process to show that. So this is the integrated processes in the drug discovery. So basically the processes divided into two. One is structure based drug design. The other one is ligand based drug design. So structure based, ligand based. What is the major difference? Only thing is if the protein structure, if the protein structure or the receptor structure. So because receptor is the general term in the pharmacological basis. The protein can be receptor, enzyme, ligand channel, nuclear receptors, and so on. So basically they are proteinous molecules. So if you know the protein structure, you can subject it for structure based drug design. If you do not know any information about the protein, but you know the molecule which is showing the potential biological activity, so in such case you can happily consider ligand based drug design. Now in our case, for example, if you ask me what is the best suitable strategy currently we are following in with respect to the anti-COVID drug discovery. So structure-based drug design is the one currently they are trying to identify. But why the success rate is very, very less? Why? Because so far, no specific and selective ligand could manage to identify with a specific receptor site, because there are n number of mutations and so many isoforms, and the catalytic domains are allosteric sites. So different complex mechanism actually leading the process in a failure way. That means we are ending up with some rational hypothesis, but when it comes to the proof of concept, may not attain significant status or progress in that. So that's how we are actually facing a lot of limitations. Otherwise, mushroom growth of publications, they are saying that this compound is very good and this compound is very good, fight against TB, that corona. Then why we are not really getting that compound into the market? Just imagine. Just if you look at the some publications in the Google, publication defines a compound as a potential compound to fight against particular disease. Just a question, why not this compound can be further modified as a drug candidate? So it's the perspective is totally different. That is, the laboratory scale may not be aligned to the industrial scale, and the laboratory methods may not be aligned to the regulatory methods what they follow in the real drug bioassay screening, and whether the bioassay screening is a standard, validated, and optimized for the special conditions, or what is the applicability of the domain. So keeping all these things in view, developing a molecule, identifying a strategy is rather very difficult. However, once based on the data, based on the data and the experimental evidence, we can further identify some related molecules. However, one thing I would like to highlight here is that last time when we were interacting with our team members, then they asked what is the validation of the computational simulations in the drug discovery. They say less than 1%. Then they might raise it that if it is less than 1%, why should we do the simulation? Okay, if you look at the success stories of the simulation, majority of the compounds identified under chemotherapeutic diseases, antiviral, anti-cancer, all based on the computational studies out of Human Genome Project. And upon successful publication of the human genome sequence and the accessibility to the targeted proteins to across the globe in the Protein Data Bank, European Bioinformatics, and so on, the majority of the people have started showing interest in developing some hypothesis. And if the hypothesis is very reliable, and if the hypothesis is considered to be valid, then majority of the companies are looking into that to adopt the published information and start up with small R&D. So this is how the process of acceleration is taking place, especially mediated by the simulation. So something is better than nothing. So instead of going through the conventional way, why don't we engage with the advanced technologies available currently? So that is how the process of transformation will generally takes place. So if ask such question to anyone, definitely we would say it all depends on the success. So in the conventional process, not even 1%, because it's a 0%. So there is no guarantee of success in either way. So in such case, why don't we consider these complementary tools to enhance our processes? So this is what the justification had given to the critics, what they say, why do we generally consider the simulation to scale up the processes like that? And I will introduce few structure-based drug design and ligand-based drug design strategies. If you look at these animations, this is protein-protein interaction and protein-DNA interaction. And DNA ligand interaction, the moment you see DNA ligand interaction, the best compounds we memorize is that alkylating agents in the anti-cancer drugs. So protein ligand interaction, majority of the compounds like angiotensin converting enzyme inhibitors, carbonic hydrase anhydrase inhibitors, and majority of the compounds, especially in anti-HIV, the reverse transcriptase inhibitors and HIV protease inhibitors are totally based on the computational tools. That's one of the strategic outcome of the simulation, because that is how the mimicking is actually been successful since some time. Starting from back in 1970s, the process accelerated since last 3-4 decades. This process has been in the R&D sector at pharmaceutical industry and biopharmaceutical, biotechnological industries as well, proteomics, genomics, and on. So I just introduce a few other concepts like homology modeling. So for example, the three-dimensional structure of the protein might not be always possible to isolate and extract to get the single crystal. It may be complicated to get the three-dimensional. In such case, you may be considering a template protein molecule, and this template protein molecule can be further subjected for sequence analysis. Based on the sequence similarity, we can do the what we can say that this is folding, because the folding of the sequence is if the sequence is more or less similar to each other, of course the protein folding also must be same. So keeping that point in view, they have projected one strategy, that homology modeling, which is mostly applies to the sequence is known but no three-dimensional structure. Yeah. So this is some information about the fragment based. This is what actually currently we are targeting the coronavirus by, because currently we have full information associated with all structural proteins of coronavirus. But however, we do not have the specific ligand which is showing the which can bind to the target site very effectively and also to inhibit the process. And this one we manage the fragment based, is that this the incremental construction of the ligand based on the bioactive pharmacophoric fragments. And these pharmacophoric fragments, when combined together to design a full molecule and see how the enhancement or any reduce, that means reduction or improvement. So if it is improving, we will further replace with some bioisotopes. If it is there, if there is any decrease in the activity, we may replace with some other fragments and check for the bioactivity. So this is all about the incremental constructions of the ligand fragments in a computational model. That means the simulation environment, and we study whether this molecule can bind to the receptor well or not. So this is all about the simulation based drug design. The fragment based drug design is one. And molecular, this is well known strategy and most widely used strategy. Even undergraduates are very well versed with this technique. Yeah, because this is very simple technique. You can actually directly download the target from your wish. Actually, you may be interested with diabetes, you may be interested with cancer, or leprosy, or any tuberculosis, any type of disease. You just identify some established drug target and search for the drug target in the protein data bank, download the molecule, and design your own molecules virtually using the online based computational drug discovery softwares, and do some docking study and see whether the compound is having some binding affinity, or how do they identify the binding affinity. It's all about the energy differences of the complex and non-complex forms. The complex form is the free energy relationship between the complex ligand before it goes into the binding site and bind, and after. What is the energy difference? That will give some remarkable standard value to identify. So this standard value must come from where? The standard value must always comes from the experimental study. So if the experimentally derived co-crystallized ligand, if you do the docking study and get the score, that is considered as the standard. So based on that standard score, because since the orientation, the conformational energy, everything associated with real molecule in solution. So what is the best way is to consider here is that we can We can take that one as a standard to uh, compare the energies of the designed ligands and with these molecules. So this is how the molecular docking simulation works. So ligand-based drug design. When it comes to ligand-based drug design, as I mentioned, if the no protein information is available, in such case ligand-based. So only ligand is available. For example, a molecule that exhibits very potential therapeutic effect, but we don't know what is the exact mechanism of action or where it is actually binding. So such in such case we can consider the ligand based, because ultimately what we want therapeutic benefit, at the same time we want less toxic, whether the compound is successfully excreted from the body or not. So in all other aspects the compound is fulfilling, but except we do not know where it actually specifically or selectively binding. It doesn't matter, because as long as the compound is safe and we are getting some therapeutic benefit, we are using for the good sake. Yeah, that's what the ligand based drug design strategy has been developed. So there are three major strategies: ligand pharmacophore based, similarity based, and 3D QSR Pro. So similarity, as you all know, similarity means what is the similar features. Mostly the similarity means the compound may have some same type of atomic configuration, same type of functional group arrangement, same type of basic nucleus, and so on. So the similarity scores may be developed based on the algorithms, and and based on these similarity scores of a template molecule, we can organize the set of molecules in the order of the ranking. So the ranking of order will gives you which one is the best one and which one is the best one. If you ask me, I may consider first three hit molecules in the computer to carry forward to the experimental study. So it all depends on your strategic plan. What is your core objective in the R&D sector? How many molecules you would like to consider for taking into the next level of the experimental study? Meaning, for example, in the computational simulation, you have identified 100 molecules, top 100 molecules. Will you be synthesizing top 100 molecules and doing the proof of concept study, or top 3 molecules only be synthesized and doing the concept study? It all depends on the strategic plan, how you are going to implement in the R&D sector. It varies from company to company. However, the basic principle is, if the compounds are similar, the activity also may have similarity. So that is what the basic thing. And pharmacophore. What's the main difference between the pharmacophore and similarity? Here, it is generalized to a specific, generalized to a chemical feature. There, they generalize it to a chemical atom, meaning that atomic based similarity is mostly they will consider. Every individual atom is taken into account. However, in this case, pharmacophore, there are different chemical features. Features in the sense basically categorized into positive ionization, negative ionization, hydrogen bond acceptor, hydrogen bond donor, and ring system, and hydrophobic atoms. These like very generalized, because the moment you say ring system, the ring system may be a heterocyclic ring or carbocyclic ring or fused ring system, any ring. Any ring is ring, like that. So diversity of the chemistry can be enhanced more in terms of pharmacophore modeling. In whereas in case of similarity, most of the times you can understand, you could see the type of molecules as similar type of backbone, similar type of skeleton, backbone skeleton. Why? Because it will take every atom into consideration. So like that. So one of the major parameters associated with the pharmacophore modeling are two. One is interatomic bond distance, and the other one is interatomic bond angle. So these two things will be considered more, and also when we take into consideration the interatomic bond distance. So here hydrogen bond acceptor is there. Here hydrogen bond donor is there. The distance between this acceptor to this donor and here ring system is there. So this acceptor to this ring system and this ring system to this donor, it makes one triangle, and this is the model called pharmacophore model. Why? Because they assumed that this hydrogen bond acceptor and donor and ring system are the key features responsible for the observed biological activity. So this observed biological activity must be retained. So in order to retain, so designing an interatomic, because there will be no changes in the interatomic bonds, and because the standard chemistry, as you know, it's established in nature. So whatever we identify the bond and angles and distances will retain the same uh, framework, the backbone. However, still you can explore different types of hydrogen bond donors, different types of hydrogen bond acceptors, and different types of ring systems. So that's how the chemical diversity further explored based on the pharmacophore modeling simulations. So moving on to the quantitative structure activity relationship. So this is one of the regulatory model. So simulation at a higher end is the QSAR. So far, whatever the simulations we discussed, there is no regulatory guidelines for that. Actually, whatever you do is actually that doesn't matter to the US FDA, as long as you need to show the proof of concept study of the compound that you have selected to proceed further into the drug discovery phases. As I mentioned, none of the drug discovery phase can be replaced. So it doesn't matter. Or suddenly, all of a sudden, you bring some compound. If you would like to enter into drug discovery phase, okay, you are most welcome to do that, as long as you pay the risk of whether success or failure. However, in the QSAR aspect, this one is well, um, what we say, it is strategized, it is regulated, and also specific guidelines developed, and there are several number of directives on how to develop a QSAR model. Why? Because based on QSAR modeling strategy, very good number of applications, not only to the drug discovery field, but also to several other industries like agriculture and chemicals and pollutants and several other diverse range of applications are there. And these two key people are responsible for the initiation of this strategy back in 1874 and 1964. And starting with a molecular descriptor. Molecular descriptor is nothing but a chemical or physical or topological or steric or electronic property. It is a property of a molecule. For example, molecular weight is one molecular descriptor. Partition coefficient, one molecular descriptor. And what we say, Andrews affinity or total polar surface area, one molecular descriptor. So these are, everything is molecular descriptor. There are different categories of descriptors. Mostly the molecular descriptor that can be generated based on the empirical calculations rather than the experimental method. Because if the empirical calculations, if you could generate the descriptor, you no need to develop any specific reactions or methods or experiments to derive such information. So most of the times the molecular descriptor that relies not on the laboratory experiments is more valuable. Yeah, because in the initial stage, we are trying to put our efforts to reduce the number of laboratory experiments and to reduce the cost associated with it and to reduce the manpower. So how can we do that? So ultimately, we may need to reduce the total number of experiments what we are planning to do. So this is one of the best book I would like to suggest, if possible. Any access is available for you to consider. So we have structural interpretation. However, the molecular descriptor must have some structural interpretation and a good correlation with at least one property. discriminate among isomers, because as you all know very well, because you are from pharmacy background, and back in 1960 around, the thalidomide tragedy has happened. By the time they know that there are two isomers which are optically active existing in the molecule, 60,000 born infants are with phocomelia, and majority of the children born infants with no limbs and joints. So deformities, teratogenicity effect because of one of the isomers of thalidomide. So why? Because at that point of time they do not know. The molecule is FDA approved molecule. So who is responsible? So it is regulated and officially released into the market, and that drug molecule created such a tragedy. That's why they say thalidomide tragedy. So meaning to say that there is no difference between animals and humans. Okay, so this is how we may need to consider safety and efficacy of the molecule is the utmost priority. So, keeping that point in view, should change gradually with the gradual change in structures, and should have the correct size dependence related to molecular size. Should not be based on experimental. Yes, of course. So, as I mentioned, data set curation. Data set curation is one of the most important property that we have to consider before we proceed for the quantitative structure activity relationship modeling. Data set curation. So what do you mean by data set curation? First of all, curation is the process of removing error. If your data is there, if the data is a error data, so and this data you are giving as an input for the machine learning to model a QSAR, and certainly whatever the model outcome from the process subjected for the simulation will be having lot of precision, and that model may not be the reliable or validated model to conduct the screening process. So data set curation in terms of both chemistry and biology is the very significant step associated with the QSAR modeling. So I will introduce a few steps here. Original set, the raw data. Once after you get the raw data, you may need to see the first chemical curations to removal of mixers and inorganics. If any mixer molecules are there, remove that molecule, and inorganics, structural conversions and screening of the salts, normalization of the specific chemotypes. What does it mean? Specific chemotypes, compounds belongs to the same category of the molecules. That means the dibenzyl type of compound, diphenyl compound. So some specific chemotype. For example, one is pyrimidine, the other one is pyrazole. For example, both are two different ring system. One is six membered, the other one is five membered. So it is not same chemotype. It may be same chemotype with respect to the heterocyclic category, but chemically, their structural resemblance point of view, they are different. Okay, removal of duplicates. If any same compound having a similar type of data, that type of data may not be suitable. And finally, manual inspection of every single compound subjecting for the simulation must be taken into consideration. And duplicate analysis, the same process whatever is taken under chemical curation, similar type of information we have also need to consider with respect to the biology as well. Why? Because the biological data is one of the key trigger that will either it takes to the good side or it will takes to the bad side. So either way is possible, especially because of the biological data. Why? Because this biological data, many of the biological assays will give false positives. Why? Because due to the proper validation, proper improper applicability of the domain, and also the standards associated with the running of the experiments and total number of experiments performed for the cross validation and so on, and misuse of statistical methods all leads to false positivity. You need that. Yes, you can see the value, very good value, but it's a false positive value. So when it move to the next level, you will understand. So that value is probably because of, and this process, I could not recall. There is a very good terms for this in the high throughput screening. Uh, in the high throughput screening, when we are doing the bulk screening, these are actually called as um, these are the ones responsible, the ligands, which will produce the false positivity, and those ligands, uh, we could very difficult to discriminate among the false positivities and positives in some types of experimental conditions. So exclusion of unreliable data sources must be the first one actually. And intra laboratory experimental validation, if you further fine tune the experimental standards, this can be actually maintained easily. And the detection and verification of activity cliff. Activity cliff, for example, 0.002 micromolar, 0.003 micromolar. So that's actually considered as activity cliff. Why? Because very close structurals, almost same activity, just a matter of 0.001. So it does not give much information, and it could not produce any distinct contribution to the further updating knowledge. So what type of knowledge you are trying to get it from that is very limited. So that's considered as activity cliff. So we may need to verify and remove such activity cliffs as well. And tuning of dataset modality index is one of the mathematical process to consider, and the consensus QSR predictions to curate mislabeled compounds. So these are all the final regulatory methods, so mathematical standards like correlation coefficient, regression coefficient, the cross validation, predicted error sum of squares. These all things will be further taken into consideration in the QSR validation. So there will be no major hurdles to get the. One of the major problem associated with it is that the getting the curated data set for model development. Most of the times you may not get a reliable, curated, chemically and biologically curated data set readily available for doing the simulation modeling. So that's the major hurdle associated with this QSR modeling. So these are the various steps associated. Once after you get the curation, remaining all steps nowadays accelerated through the softwares. Previously in the two-dimensional QSR modeling, those times we may need to use the manual methods, but now it's totally automated. There are good number of softwares are available, both commercial and open source softwares as well, to develop some QSR model and validate. So for developing which model, the curated data set you are using, that is generally called it as training set. So if from the training set itself, if you take some molecules and validate this one, that is considered as internal validation. But if you consider the molecules which are already available in the external databases like Sigma or Reach or any commercially available ligands, and you do the screening for this and predict some activity and carry forward to the experimental conditions, and this is considered as external validation. So external validation is the key validation. Internal validation, many of the times, majority cases, it will get successful. But the external validation and proof of concept validation is the main, as I said, the valley of death. The value of that is the external validation and proof of concept. Once if these two things are successful, so the QSR model generated is robust, valid, reliable. So we can integrate this model to a specific domain, in the sense this QSR model can able to predict anti-diabetic agents PPR gamma as agonist from 1.0 micromolar to 3 micromolar range. So this is what the applicability of the domain. Above that and below this, not suitable. So this how the specific applicability of the domain is the very important one. So these are the 5 major OECD principles developed for the standardization and regulation of the development of the QSR model. So what is OECD? Organization for Economic Cooperation and Development. So this mostly the animal testing and all OECD principles generally they follow. For animal testing, acute toxicity studies, like I think if I'm recall correctly, OECD 425. I think for the acute toxicity studies, but I do some acute toxicity studies back during my PhD. So I do remember that. So OECD guidelines are the some regulatory guidelines to develop and validate the model as well. So these are the 5 principles we can consider. The first one is a defined endpoint. That means the it should not be like ambiguous, like where we are actually ending this one. It should not be a continuous process or any side reactions associated with it. It should end with at particular point. For example, the reaction started enzyme, for example, consider the enzyme kinetics. At one point of time, the moment you add um, the acid, the enzyme will stop. Okay, the enzyme further inhibited. So some definite endpoint must be there. That is the first principle. Unambiguous algorithm, that means on what basis this algorithm is actually working. So developing a QSR model, whether this model developing based on the standard conventional theories, empirical relationships integrated into algorithm, or this algorithm is directly is the a replica of some other thing with the hiding codes and all. So total programming or background working of that algorithm must be disclosed to the uh, members. So that is one of the thing. And first one, the principle 3, a defined domain of applicability. As I mentioned, the domain of applicability is one of the most important as well. And appropriate measures of goodness of fit, robustness, and predictivity. Almost the same like analytical method validation. Here also the goodness of fit, linearity, robustness in multiple different types of softwares, and predictability. What is the range these compounds can be identified? So all those things. Mechanistic information if it is possible. So mechanistic interpretation, if it is not possible, it's not uh, it's okay. Still, it can be considered because it is just optional. But if you can get the mechanistic interpretation on how it is actually showing within the binding site region and all, further that is the 7 dimensional quantitative structure activity, which is high end QSR modeling. So that is currently not required for every model. So these are some theoretical aspects. You can get this information totally directly from the OECD guideline. Just you can press OECD guidelines for QSR model development. You can all get this data. Some important points highlighted. So one of the major errors affecting are this one, use of inappropriate endpoints. OECD principle 1 will be affected. So your bioassay must have appropriate endpoint, so that, and incomprehensible descriptors will also affect the, like that you can see too narrow range of endpoints, misinterpretation of statistics, lack of mechanistic. So OECD principles are mainly affecting because of these errors. So try to limit these errors when you are trying to develop a QSR model. Keeping these points in view so that all the OECD principles can be satisfied and further um, uh, can be subjected for getting good model, and we can validate the model as well. So the main parameters considered are reliability and reproducibility, critical issues in the simulation experiment. So this is one of the example for atom-based 3D QSR model developed at International Medical University. So coming to applications point of view, as I mentioned, this is the regulatory model. QSR can be adaptable at every stage of the quantitative drug discovery, design, and development. However, the main problem is associated is the data set. Statistically significant data set only can produce a reliable model. So n is equal to not less than 15 molecules for as a training set, not less than 5 molecules as a test set to, to do that. So, but it can be adaptable based on data. If the experimental data is readily available, simulation model, the QSAR simulation model can be developed using sophisticated algorithms, the 3-dimensional, 4-dimensional, and 5-dimensional. Uh, so some success outcomes of QSAR in drug discovery includes these are some of the example drugs. Very limited examples I provided, but a very good quantum number of success stories are actually present for this category. So in conclusion, summary, what I would like to highlight is that computational simulations playing major role in the development of therapeutically important small molecules over 3 decades of several FDA approved drugs designed by intense use of various types of CADD simulation strategies. As I mentioned, there is no perfect flow for the CADD strategies. As you are the computational chemist, as you are as a practitioner of the chemoinformatics and bioinformatics, you can design some workflow and try to implement and see whether the workflow is integrated between SBDD, LBDD, and FBDD, or FBDD and SBDD, or SBDD and FBDD. So like that, it's all about the way how you design the things and you implement in the process. And this workflow, whether it produces a hypothetical compound, once it gets successfully approved by a proof of concept experimental study, and this workflow may be adopted across all the industries globally. So, however, it may be an intellectual property as well, because um, that could be the successful workflow in that particular R&D sector. Okay, the real breakthrough. Uh, this is, in my personal opinion, however, the global perspective as well, we can, science will come when we can use only simulations as the reliable tool to ensure that only active designs are used for scaling the processes. So meaning to say that once you do simulation, whatever the outcome is there can be readily transferred to the next stage. That is the situation we are expecting. That is the real transformation and real advancement we actually come through. And this really the global outbreak, if that's the case, in the advancements happens. Okay, our take home message is just to stay calm and use a simulation. So these are the references used to construct the slides. My special acknowledgements to Associate Professor Dr. Muhammad Zulkifli, Dean School of Pharmacy, IMU, Malaysia. Associate Professor Dr. Gansuke, Associate Dean, School of Pharmacy, IMU, Malaysia. Professor Malika Chandra Pijka, Associate Dean, School of Pharmacy, IMU, Malaysia. Dr. Lee Choisin, Head of the Department of Pharmaceutical Chemistry, School of Pharmacy, IMU. Professor Dr. Vairajendra Prasad, Andhra University College of Pharmaceutical Sciences, Principal, India, my research supervisor and director, and Professor Dr. T.V. Narayana, National President, Indian Pharmaceutical Association, India, Professor Dr. S. Vijayadhara, Chairman, Indian Pharmaceutical Association, Education Division, India, and Dr. R. Srinivas, President, Dr. C. N. Srinivas, Secretary and Correspondent, Chetrolio Hanamaya Institute of Pharmaceutical Sciences, India, Colonel B. Venkat, Director, Faculty Development, All India Council for Technical Education, New Delhi, Professor Vijay Vaidya, Executive Secretary, Indian Science Technology Education, Technical Education, New Delhi. Associate Professor Dr. Venkatrao Utla, my co-scholar and my best friend, who actually initiated the communication and networking between the Cheprolani Institute of Pharmaceutical Sciences and IMU, India, organizing members and beloved participants, press and media. Thank you all for your um, time and also the patience and listening to my speech all the way since last 1.5 hours duration. Thank you all once again. I'm ready to take any questions from the participants now. Over to the host.
So participants can unmute themselves if there is any questions. So I think there are no questions.
Okay, thank you so much once again.
Thank you.
See you, Dr. Pramod, in the next event.
Yes.
Thank you, all participants. Have a nice day and stay safe.